Right, lesson 1, natural selection theory. Here are some of the things that you will need. Can I move myself around? Look at that, excellent. You need time to do it. All the usual notepads and stuff, textbook and your workbook. You've got copies of the spec. That's what we're going to cover. Myself down there, out of the way. And a bit of a recap to start with. 3 things that you've done in AS. Time to pause on these 3 things. Let me go through them. Transcription and translation and protein synthesis. Well, we've got DNA with your nucleotide bases on. That's your genetic code. That gets transcribed onto single-stranded messenger RNA. So we've got triplet code, and then we've got that triplet code transcribed into codons in the nucleus. Then the messenger RNA moves out into the cytoplasm, and at a ribosome gets translated, and various tRNAs with different amino acids in will get matched up, codon to anticodon, and you'll get your polypeptide formed with peptide bonds between the amino acids, and that's your protein. That requires energy, so it's just a little recap of protein synthesis. The other second thing that we asked you about was mitosis and meiosis, because these will be useful. So we've got mitosis down here on the left. First thing that you notice is that what we start with is what we end with. So it's essentially just a copying of our original cell. It's going to be genetically identical, apart from we are just making 2 daughter cells from our original cell, and we go through the stages there of doubling up the DNA, so DNA replication, and then separating those 2 chromatids, and then we've got our new sets of chromosomes. In meiosis, if we look at what we start with, so a normal body cell, as it were, and what we're ending with, you can see there's 4 chromosomes in our start, and there's 2 chromosomes in our end cells, our daughter cells. So we're halving the number of chromosomes. And these, and you maybe can't read the letters there, but sometimes represented in colours, these are genetically different due to lots of things going on here, like crossing over in prophase. We've got independent assortment or independent segregation of the homologous pair. We've got independent segregation of the chromatids. So these are going to be genetically different. And of course, we've got four cells in the end. So these are our gametes, our sperms and eggs. And another source of variation is in mutations. This is just a really simple version of this. We've just got one triplet code, CAT, giving histidine amino acid. And these are all the same. So we just showed you one changing there. So from CAT to CCT, which is giving proline, which is a different amino acid. And that may or may not lead to a malfunctioning protein, or it might lead to an improvement in the protein, but certainly different. Here's an example that we'll chat about. We'll maybe come back to this in a later PowerPoint. It was a classic natural selection example using Californian salamanders. So what we've got to start with, we've got a population here at the top, and they show a variation of phenotype. So phenotype is what characteristics does an organism have. It might be what it looks like, or what blood group it is, or that sort of thing. But what characteristics are evident. The genotype are the alleles present, and the DNA and that sort of thing. But the phenotype is what it looks like, what it is, what it does, that sort of thing. Genetic variation and environmental factors will affect the phenotype. So we've talked about mutations that will change the DNA, and so that might change the polypeptides or proteins produced, which will affect the phenotype. We've talked about variation occurring in meiosis in the past slide, and of course random fertilization of gametes. Males will produce millions of sperm, and only one will fertilize the egg. So, and all of those millions of sperm are all genetically different. So there's another factor that increases variation. Environmental factors, things like food and disease. Classic example, identical twins. If you feed one identical twin and then don't feed the other one, then they'll have a different phenotype that you can see. I want to be bigger, I want to be smaller. Disease, accidents, predation, things like that. So, things happen to change the phenotype of organisms, and that's not genetically related at all. And so here in our little diagram, we can see different phenotypes. These are all essentially part of the same species, called the ring species, where they're all slightly different to each other, and on the natural selections occurring, and they're on their way to becoming separate species. But technically, most of these can interbreed. Remember that we're using a definition of species, which is ability to interbreed and produce fertile offspring, which most of these can do. There's a couple of examples where they'll produce a hybrid that isn't very fertile. So we'll go back to that salamander example again, but let's just throw it open to you again and think about plants. Just hammer home this environmental factors. How can they affect things? So how does the phenotype of plants be affected by environmental factors? So how could that change it? Press pause for this video and spend 2 minutes thinking about that. Okay, hopefully you've had a good think about the things that can affect plant phenotypes. Hopefully you came up with things like light, water, ions. So more light, more photosynthesis, more growth. If you cover up a plant, it'll die. Will certainly not grow very well. Water, you need to water your plants at home, else they won't grow. And ions, things, like metal ions like magnesium needed for chlorophyll, nitrates, ions, things like that. So organisms can be affected either by their kind of genetic variation or their environment. So here we go with a natural selection. Here's our Californian salamander, and up near the top where the ancestral salamanders were, these are quite varied in appearance. Our natural selection points that we'll talk about. Salamanders, there's overproduction of salamanders, so the parent salamanders will produce lots of offspring, And during their lives, lots of things will happen to these salamanders, which will mean some survive better than others. So the obvious things we normally talk about are predation. Do the salamanders get caught and eaten? Disease. There's diseases prevalent in wildlife, and competition between the salamanders. So there's competition for food, competition for not being eaten, and things like that. So not all the salamanders are going to survive, and that's what that word differential survival means. Some will survive, and those that survive, presumably they will go on to reproduce. And so those organisms with the advantage, they survive, they reproduce, they'll produce more offspring, and they will pass on those alleles that made them favorable. And so if there's an allele that made it less likely to be eaten, that will get passed on to the next generation. If you don't survive, you don't reproduce, and you don't pass on your alleles for whatever it is you are currently looking like. And then this differential differential survival, differential reproduction, has a change on the allele frequency, which is how many, what proportion does each allele have within the gene pool. So if there are more alleles for being red or black in that gene pool. So with our Californian salamanders, some of you might have seen this video. I've actually put a link to someone who studies these and can give a much better version of this story than me. However, up here, this is where the salamanders started, 20 million years ago, something like that. And there's variation in these. I think the forests at the top, and the variation in the salamander phenotypes. And then the salamanders wander down southwards, and they can either take two routes. There's a coastal route, and there's a kind of inland route with a valley, the central valley in between. And salamanders won't go. It's too hot and dry in this central valley. And on the inland side, then you can see we've got a change in the phenotype of the salamander. For example, this one becoming, ending up a cloudberry one, where it's very blotchy, and this is to avoid being eaten. So it's camouflaging against the background to avoid being eaten. And so down here, here's an example of natural selection. So there's lots of mutations and meiosis and that sort of thing. Some will have different coloured blotches. The ones with the most camouflage blotches will survive, reproduce, pass on those alleles for those particular blotches onto the next generation, and the allele frequency for having those blotches will increase. If the salamander is plain, then they're easily seen by the predators. And so alleles for being plain, they won't survive, they'll get eaten, they won't pass on those favourable alleles. Same for army uniforms, they're all kind of blotchy, to cut out the edges where you can spot things. Whereas down here on the other side, the coastal side, you can see salamanders are becoming one colour, but quite red. And so what's happened there is there's a poisonous newt that advertises its toxicity by being bright red. And these salamanders are mimicking this poisonous newt, so they're tending to be this poisonous newt. And so what happened is, here we've got some kind of normal-looking salamanders. Those that might have a mutation to give red skin, then predators won't eat them because they think they're these newts. And so these will survive, and they'll pass on the alleles for this red skin. They'll pass it on to the next generation, those favorable alleles, and the allele frequency of those red alleles, as opposed to, say, blotchy alleles, will increase in the gene pool. And in the last couple of lessons in this topic, we'll talk about evolution, but really this is this natural selection that leads to the evolution when these can't interbreed with each other. So that's natural selection in action. Let's just talk a little bit about Charles Darwin. So we'll talk about Charles Darwin and evolution. There's Charles Darwin. You usually get a kind of big beardy version of him, but he was when younger when he actually did most of the voyages and the work. There's a finch, and 31 onwards you can see the trips that he made, and these are the Galapagos Islands. And as you can see here, there's lots of islands, but they're kind of separated, and so some animals, they might have been in one place, and they've spread out over time over the different islands. These islands don't have the same climate, same wind, rain, and so on, and food sources, as influenced by the environment. And so there's different conditions on these, just like the salamanders down each side of the valley, different conditions that lead to different finch beaks. So there's the beak of the finch, and just time for you to have a little think about what do you think these beaks will look like if these finches were eating kind of big, hard seeds. So if you try and eat a seed, sometimes quite difficult, or catching little insects. What do you think the beaks would be like for catching little insects? So 5 minutes to have a think about that. Right, hopefully you've had time to think about these beaks, and you might have got something a little bit like this, where there's our ancestral ground finch. So that's our starting position, and you can see how this is actually called adaptive radiation, where we get spreading out from our original organism. But you see some that eat insects, some of them have got quite pointy beaks for finding little insects and so on. It depends on exactly what they're eating. The birds that are eating, for example, big buds and fruits, they've got a big beak. And birds that are eating seeds, and so things like this, for big strong beaks for getting into these hard seeds for the food. And so, here's the example again. So, for example, this beak here, birds with the mutation or genetic variation with a slightly bigger beak are better able to find food from the seeds. They're more likely to survive, more likely to reproduce. They will pass on their favorable alleles for having a big beak onto their offspring, and the allele frequency for the the big beaks will increase in that gene pool, in that population. And so eventually, nearly all the birds will have beaks, in that particular ecosystem. So I think you've this a few times in this point. So just to recap, individuals show variation, and usually parents will overproduce, so not all of their offspring will survive. That's that word, differential survival. Those at the selective advantage pass on those alleles, and that affects the allele frequency over time. So in the next lesson, we'll give you 3 examples that you need to fill in a table in your workbook. I'll give you some teasers for things to say in the workbook, but not give you all the answers, or we'll put those in a separate video. And we'll think about the assignment book questions eventually as well.